The interface we propose for Cell Databank has two main components: a WebGL interface up top in the style of Google Image Browser, and below this is text content in the style of a MediaWiki. Ideally, we also want to be able to resize or hide one or the other so that we could make either of these full screen. On the first page, you'll notice we can pick between a few different model organisms, which would be slowly added over time. So let's say we're interested in mouse. On clicking an organism, it begins to load the model. Notice also that the page below has changed and gives us information about this organism. Up top is basic information telling us about the average size and weight of a mouse, and so on. And below that we have dataset information, which will tell us information about this specific model and how it was acquired and by who. Building on the Zygote Google Body Browser, now an open source project called Open3D Viewer, the slider on the left provides an easy way to fade out the different systems in the mouse so we can view its anatomy. If I'm interested in a particular component, I can click it and see these important options. The first is to load a more detailed model of the mouse. Below it, Load in Place does the same thing in loading a more detailed model, but does so in place. And Calculate Size would tell us the size of this particular brain mesh. So let's load the brain now in a new coordinate system. Here's the brain, and we see the slider now reflects the different parts of the brain which we may want to show and hide. In actual fact, there are hundreds of parts to a brain, so if we want more fine-grained control, we can click here to shift to a list view. In this more advanced view, we can individually select, show and hide the individual compartments or groups of compartments. Below this, you'll notice a list of cells. This represents a list of all cells you'd expect to see in the brain, but the ones at top in black are linked to 3D models which have actually been docked. Notice the little purple dots represent our cells, but since we can never pinpoint these precisely, the yellow sphere represents an area of deviation and helps us see where the cells are as well. Again, we can select any compartment we choose, so let's click I if we wanted to add our own cell to that compartment. Positioning cells in 3D is very difficult, however, so this is why we reuse a familiar model from Google Maps, where we can change between different views. Currently we're on model view, but let's switch to slices. Notice we can view the top, side or front views, words which make a lot more sense than transverse, sagittal and coronal. In the slice view we can see slices of raw image data on which each model is based, and here we can help identify coordinates and the appropriate location where a cell comes from. If we wanted to scroll up and down slices, we could use the up-down arrows or this slider here. Meanwhile, the hybrid view will show us our last visited slice in the context of the model, and here we see a parallel orthogonal view, which allows us to better measure distances between any two arbitrary points by this grid, something which doesn't really work in normal perspective view. Let's actually look at a cell now. Let's select and zoom up on the optic chiasm, and there happens to be two cells here. Currently the cells are represented as oversized spheres, but if we click, we can load one. Notice here we can see its unique model ID and also its rotation relative to the brain. Keep in mind, rotation may be roughly known for some cells, but in many cases a rotation will be completely unknown, so that's something else we want to reflect in metadata. Here we load a cell at its original rotation. We can see the compartments which have been segmented, and that our purple plasma membrane defaults to semi-transparent, so we can see inside. In this list, the little blue mesh icons represent a mesh, while the expand arrows means a group of meshes. So here we can actually expand mitochondria and actually see that there's a bunch of mitochondria, each with a unique number. Something I want you to notice here is that the cell, organ and organism level views are all very similar. The only real difference is the scale is different. Just like before, however, we have the option here to look at the raw data. So let's flip back to the easier slider view though. Different compartments will be automatically matched to different layers so that they can fade in order, and how we peel back the plasma membrane, mitochondria and so on. This interface thus provides a very nice way for the researchers to show off their cell models, but don't forget we also want to make this program more useful, more than just a visualization tool. As such, we can actually select and right-click a group of mitochondria to graph the length or calculate size. Let's click Calculate Size, and we can see here the number of mitochondria plus their total size, surface inflammation, and so on. If we wanted, we could also click an individual mitochondria to work out its size. This information is starting to get more useful, but what makes this interface really useful is that below this, people have been allowed to enter information about the average mouse dentate gyrus neuron. This page here represents a wild-type mouse, and the way it works is that all these fields start as red and unknown, but people who visit can fill in the blanks and provide at least one citation for each piece of information. As someone who studies pancreatic beta cells, I may not know about the size of a synaptic vesicle in this type of neuron. Probably I don't. But there are scientists out there who will actually study and specialize in the size of these vesicles in particular types of neurons. And so over time, we can help populate this information and see where our gaps in knowledge in quantitative cellular information still lie. Here we can see that someone has entered the average volume for a mitochondria in this cell type, and if we want, we could chase up the literature and see how stereology was used to calculate this. In the case of brain cells, it's actually very difficult to calculate total volumes because they have such long axons and large complexes of dendrite. Indeed, if we read about this data set information, or even look closely at the model itself, we'll see that although the main body or soma of this neuron is captured, we'll often be missing a lot off to the sides. Ideally, a cell is captured in its entirety, so it can be fully analysed as a unitary complex system, but in the case of neurons, it's really not very possible. Let's zoom out now, and you'll notice that if I zoom out far enough, it presents a link to go back to the next level up. Also, I could actually click any of the breadcrumb links below if I wanted to jump back to the mouse or home screen. Let's now return to our mouse and type in the word pancreas. The Open Viewer has this neat interface where you can jump to any object, even make use of synonyms, useful for objects like the cell's plasmamembrane, which may also be known as the cell's membrane. Just to be different, let's click Load in Place so that we can view a more detailed pancreas model while we can still see the mouse. There aren't many parts of pancreas, but we do have a couple of cells here, and also a little bit of a colour scheme whereby the purple cells represent wild type, the red ones may represent, say, a diseased cell. Let's load this beta cell here. This is a wild-type pancreatic beta cell which is captured in its entirety at high resolution, and once again we can peel away the layers and can see what types of compartments have been segmented. Notice here I've shown a lot of unknowns, but in actual fact there's hundreds of studies and hundreds of experts who will know these pieces of information. People study all types of different cells, not just mouse cells, but organisms like E. coli, zebrafish, monkey, plant cells, Drosophila, and everything else you could imagine. Here I wanted to demonstrate something a bit fancy, and that's the idea of comparing models. The crudest way to compare models would be to open two separate browser windows and compare side by side, but here I wanted to talk about the possibility of a split OpenGL window. So I'm going to grab the separator on the right and drag it across, and I can see two views, identical at the moment. Now let me zoom out back to the pancreas and open a different cell. Now suddenly I can view these side by side. Currently whatever I do on one side is independent of the other, but what I really want to do is to view these two cells at the same scale. So I click this lock scale icon, and it wants me to switch to an orthographic view, which is fine. Now when I zoom in and out on one, the other stays at the same scale. We also have an icon to lock actions. So with this click, actions like changing rotation or opacity are also locked between the two cells. So here I've demonstrated the comparing of two pancreatic beta cells, but one of the neat features of this is that it would make it easy to compare the size of structures of different cell types, or perhaps even the difference in anatomy between two different animals, or maybe the same cell from two different animals. Some of the final buttons we can see in this interface is this one to flip the left and right. This button can show a little information about the model itself. This one brings up some basic help, so we know what the mouse buttons do and how to navigate. This button is for settings. Also, it'd be nice to have a button to bookmark or show someone exactly what you're looking at, a bit like in Google Maps where you post a link. One useful button here is to either upload or download a model. Downloading a model itself is pretty explanatory. It would come down as an OBJ or VRML format. To add an object, it should be required, however, that you first upload the cells or, say, mouse model to a database like the Cell Center database, including both the model and the raw image data itself, so that we know that it's a real cell. And also, you want to make sure all the metadata, like specimen type, condition, age, and so on, is all recorded prior to finding where to dock this cell or organ inside its organism.